Good evening and welcome. First, I would like to give my sincere appreciation to the organizers for inviting me once again this year to present our work at Pep Talk. In today's presentation, I will focus on how we were able to use a variety of structural mass spectrometry tools to investigate the binding of a bispecific antibody. I hope you will have an appreciation for the tools and techniques presented today for understanding the higher order structures of antibodies entering cell engineering and early development. Here is an outline of my talk. I will first give a brief introduction to my group's role in large molecule early development. Next, I will describe several different tools and techniques we call structural mass spectrometry that has enabled us to understand antibodies that have a propensity to aggregate. Finally, I will conclude my talk by summarizing our findings and give you a peek to our future. This figure represents the general stages in large molecule early development. Once a target is declared during pre-portfolio research, candidates in the range of 100 to 1,000 molecules get screened and filtered through lead generation, lead selection stages to less than a few dozen molecules that enters our department that was recently renamed as the Cell Engineering and Early Development, or SEED for short. The SEED group is responsible for the selection of the cell line and early development activities, and is a function that sits in between discovery and development. The analytical function within SEED uses mass spectrometry and biophysical methods to characterize molecules generated during cell line selection. We work on a range of different molecules. You can see on the top left-hand corner that includes the typical MABs, drug conjugates, vaccines, but we have recently experienced an explosion of multi-specific MAB formats that kind of dominate our pipeline. Here I'm showing the different stages in a typical cell line development process. The process starts with the transfection of an expression vector with the genes of interest using single-cell cloning. These single colonies are then expanded to 96 deep Pearl plate screens, 24 deep Pearl screens, and finally end up in amber bioreactor screens, amber 15 bioreactors. The entire process takes 4.5 months from transfection to selecting the manufacturing cell line. We usually have a dozen such programs that go through these stages at any given time. So they're kind of running in parallel most of the time. And despite the explosion of molecules and short timelines, we deliver a comprehensive characterization package of our lead cell line using fit-for-purpose analytical methods, which are used for product quality assessment. For example, the table on the right-hand side shows a battery of assays we run for in-depth product quality analysis, usually at the final amber bioreactor stage. While high-throughput screening methods, such that are shown here, are more suited for triaging clones in early 96 deep Pearl or 24 deep Pearl stages of screening. In addition, the transfection pool material can also inform us on molecular properties, such as the antibody's propensity to aggregate. And analyzing transfection pools gives us enough lead time to develop additional assays, including what I'm going to talk today, the higher order structures of a molecule, if we have enough time to get that information before cell line selection. This brings me to the main topic of my structural MS tools and techniques that enable the determination of higher order structures of antibodies and antibody complexes. The topics I will cover are listed here. First, I'm excited to show our fully automated pipeline, which has been a key success stories in our lab's ability to deliver on the rapidly growing number of programs. The end-to-end automation includes sample preparation, data acquisition, data analysis, and aggregation happening in a seamless fashion with minimum touch time. As you may note, we have bridged sample preparation via automated robotic liquid handling, cold storage with mass spec data acquisition, which you rarely see in most labs. Once the data is generated and stored on our servers, a watcher automatically sweeps the data to an automated module of the Biosphere Enterprise, which is a software that resides in the Amazon workspace that centralize all the data processing on an analysis server. The application server interfaces with BIOS client or a web client for multiple users to access, reprocess, and upload analysis results. Our current workflows include both peptide mapping and intact and subunit mass analysis. The results can be automatically exported and modifications can be jointly reported. Finally, data aggregation can be performed with molecular information with external programs such as gene data. Currently, most of the data analysis for higher order structure I'll be showing today are not a part of this workflow and require specialized software tools. However, as we gradually evaluate and add new capabilities to this workflow, BIOS will be able to provide both primary structure as well as higher order structure analysis in the near future. Here's our current native mass spec workflow, which we have recently added to the Biosphere automation module. The native MS spectra are automatically deconvoluted along with their fluorescence tracers, which allows for both peak identification as well as quantitation. Here an example of a native SEC-MS analysis of a trispecific antibody that is composed of 2 scFv arms and a single Fab arm. With long separation times, we observe tetramers, trimers, dimers of the antibody, as well as fragments of the monomer and clip species and other mispaired chains quite well with a UHMR mass spectrometer, which is going to be the workhorse for almost all our native MS experimentation. So native MS in combination with ion mobility, which is a gas phase separation method, is important for the deep characterization and structural analysis of antibodies. We're just beginning to learn its importance in the analysis of multi-specific antibodies. What you're seeing here is a native mass spectra obtained with extended mass range of Agilent 6545XT QTOF mass spectrometer. So in this example, we are using time of flights rather than the Orbitraps, which is typically used for native mass spec workflows. And a time of flight in this case is sufficient with its extended mass capabilities. In this example, what you're seeing is a bispecific antibody made up of a scFv arm and a Fab arm. And we believe that the scFv domains are quite sticky, and it is known for many prior studies in the literature that scFvs can cause aggregation. Here we see both the dimer and the trimer of the bispecific antibody. And in order to further understand what domains are causing aggregation, we can do selective digestions and examine digestion products of these dimeric species. What you're seeing here is fabalactic enzyme cleavage of the hinged disulfides taking place mostly on the Fab arm. And we obtain the heavy chain 1 plus the FC fragment and a Fab prime fragment. The digestion efficiency of the scFv arm is rather poor, as you can see here, but we do get a minor scFv fragment along with the FC fragment. This is what we see when we run SEC-MS. The top is a fluorescence trace of the chromatographic separation of the dimer of the heavy chain 1 plus the FC fragment. If we now look at the leading and the trailing edge of this peak and run through an ion mobility technique known as cyclic IMS, you can actually get structural information from the mobilograms. So these are gas phase separation traces. You get a more compact structure on the leading edge and a more elongated structure and a less compact structure in relative terms, and also the compact form on the trailing edge of this dimer peak. So these are substructures that we don't necessarily see in solution, but we see them in the gas phase. So now, if you know that the scFv domain can also complex with the heavy chain 1 plus the FC fragment that we generated through a fabalactica digest that is floating freely in solution. And we form the dimer between these two fragments, and the SEC fluorescence trace shows the dimer as two distinct peaks. We can actually label them in the bottom trace. The peak 1 and peak 2 are the corresponding XIC profiles of these fluorescence traces. And if you look at the ion mobility or the mobilograms, peak 1 is composed in relative terms a compact structure along with the elongated structure. We believe that this is a gas phase artifact where the elongated structure is interconverting to the compact form, and the peak 2 is composed predominantly of the compact structure. So this is the kind of information you get combining native mass spec with ion mobility. We also collaborated with mobile ion systems to examine these subunits using high resolution ion mobility in a technique known as structures for lossless ion manipulations, or SLIM. In this example, we were able to see substructures of the Fab domain, which is not as flexible as the CFE domain. And we see compact form and a less compact form or relatively elongated form of the Fab. So this is kind of an interesting result. And on the right-hand side is the figure, is an example where SLIM, or structures for lossless ion manipulation, was used to look at antibody drug conjugates. This was a collaboration with the Pacific Northwest National Labs. And this study, we were able to obtain the DAR values of the heavy and light chain independently, and we were able to derive the average of the DAR values much faster than the conventional SECMS approach. And the values that we obtained using this very fast method in less than a minute was comparable to the SECMS results. Next, we perform footprinting experiments. So we have both hydroxy radical footprinting as well as HDX. And we use these methods to look at the binding interface. The HRPF-MS work was a collaboration with GenX Technologies. And all the work I will be showing today was performed by Dr. Emily Shea, who's seen in the top left-hand corner in this picture. Now, to give a background on this technique, hydroxy radical protein footprinting, or HRPF for short, relies on irreversible protein hydroxylation in combination with MS to provide a unique and powerful approach for high-order structure analysis. Hydroxy radical reactions are an established means of labeling, as they react with 19 of the 20 amino acids, and the only residue that does not label is glycine. This provides excellent coverage of the protein sequence, and a variety of techniques have been used to perform HRPF. Perhaps the most widely used approach relies upon the fast photochemical oxidation of proteins, which is known as FPOP for short. FPOP for short, that generates hydroxy radicals using a single high fluence short pulse of UV light. So it uses a laser. The reaction of hydroxy radicals as exposed to amino acids typically results in a net insertion of 1 oxygen atoms into amino acids, and multiple such amino acids are oxidized simultaneously. Hydroxy radicals are short-lived, and when generated by a 20-second nanosecond UV laser pulse, reactions between the amino acids and radicals are completed before any conformational changes can occur of the labeled protein. So there is no denaturation taking place during this process. In this particular technique, a variant of this FPOP method known as FOX does not use a laser. And in this technology, we are using adenine as an excellent radical dosimetry internal standard. So upon irradiation, in the presence of hydrogen peroxide, adenine loses UV absorbance as shown in the blue plot above. So when irradiated with UV light in the absence of peroxide, its UV absorbance spectrum is relatively unaffected as shown in the green trace. So when we do FOX laser-free FPOP workflow as depicted in this figure, the effective hydroxyl radical load is assessed in real time by monitoring the change in this adenine UV absorbance. Should the change in absorbance be less than the desired target, in other words, insufficient hydroxy radical load, the flash energy is increased until such time the target change in adenine UV absorbance is achieved. real time. Once we determine the radical load of the bispecific antibody monomer, we do the similar experiment to ensure we do not have any background scavenging as shown here. So we can actually do a comparison between the protein and in the presence and absence of the ligand. After we do this radical dosimetry experiment, we are now ready to perform the actual HRPF experiment. This animation shows a typical workflow for HRPF footprinting. Typically, a pairwise labeling experiment is performed where an investigator wants to learn of the difference in solvents accessible domains of a protein under different conditions. In this slide, the higher-order structure is evaluated for a purified monomeric bispecific antibody and its purified dimeric form, which is composed of two monomers. So the top is the monomer and the bottom figure is the dimer. In our experiment, the binary comparison is made between the monomer versus the dimer of this antibody. Both the dimer and the monomeric forms are then labeled using HRPF, where hydroxy radical attack decorates the solvent accessible residues. The labeled proteins are then denatured and digested and subsequently analyzed using LCMS. The differential analysis of the oxidized peptide is performed, and those domains which become protected are no longer solvent accessible, as well as those domains which became deprotected can be measured using these chromatograms. The results are then constructed in the context of the protein structure to create models which demonstrate protection or putative binding site, as well as deprotection or allosteric conformational changes. Here I'm showing the HRPF-MS results processed and reported by Foxware. On the right, we have a volcano plot of tryptic peptides covering the heavy chain 1 of this bispecific antibody. The y-axis shows the negative log of the p-value, which it gives us the confidence of the x-axis, which gives the log4 change for dimer to monomer ratios. If there is a downregulation, where the x-axis is a negative value, the average peptide oxidation of the dimer is low, and hence the dimer formation protects the peptide from oxidation. So the peptides shown in blue are both significant in their p-values, as well as the fold change is greater than 2, which we typically use as the cutoff. The plot on the right-hand side is a bar graph showing the average peptide oxidation for all the peptides that we observe. The red asterisk on the top of each of these bar graphs in the peptides that we've listed here are statistically significant differences in their ratios. And the purple rectangles showing these sequences are peptides that are common with heavy chain 2. We next map these to the heavy chain 1 structure as shown here. We get good coverage for most of the sequence, but we were disappointed to not see coverage of the linker and part of the variable heavy and variable light regions of the CFP. The poor coverage we attribute to not observing oxidized peptides that we believe was not retained in the LC column due to their small size and hydrophilic nature. In order to circumvent this issue, we took a more unconventional approach here. Instead of looking at oxidation, we examined the unmodified peptide counterparts, which are usually retained better in the column, and its change in intensity then can be used as a proxy to estimate the amount of oxidation indirectly using this equation. With this approach, we now get better coverage of this region. With all this information, we can now map this to our bar plots and volcano plots and visualize and obtain more comprehensive coverage. But you may know that the fold change differences of these indirectly derived regions do not show statistically significant differences. The p-values are low, and its reproducibility is not that great. So we can now use all this information and do paratope mapping of the protected and exposed regions of the dimer as shown in this cartoon. And as shown in the region, there's SCFE domains that may be involved in direct interactions. You may note there are small patches of VL-VL, VH-VH, as well as linker-linker interactions taking place, where the dimer regions are protected, the oxidation is less compared to the monomer, and solvent accessibility is impaired in these regions due to direct interactions. If you know that there is also a certain region that is slightly red, which is deprotected in the dimer relative to monomer, which we believe is due to unfolding of this region due to allosteric effect. We can also look across different chains. What we see here is a similar analysis performed for heavy chain 2, which has the Fab binding arm, which is more compact. We know most peptides do not show statistically significant fold changes, and the peptides that do show changes also are very low in overall average peptide oxidation. Taken all these data together, we believe that these regions of the SCFE arm are protected from solvent accessibility due to SCFE-SCF interactions driving the dimerization process. Finally, I would like to briefly go over the HDX-MS experiments we did on the same bispecific antibody. All the work I'm showing was performed by Dr. Yoshi Hamuro, who is our local expert at Janssen on HDX. In a hydrogen deuterium exchange, what we monitor is the hydrogen to deuterium exchange reaction on the backbone amide, which is very, very slow. This picture illustrates the steps of HDX workflow. We start with a hydrated protein in solution, where the blue dots are the backbone amide hydrogens. When we mix our protein of interest with a deuterated buffer, these backbone amides hydrogens gradually exchange for deuterium. After a certain exchange time, we are going to have a partially deuterated protein, and then we can stop the exchange reaction by lowering the pH or the temperature so you can quench the reaction, as seen where the colors of these dots become a little shaded. We next digest the protein, usually using pepsin or some other acid-active protease to generate peptides that we inject and perform LC-MS. This mass spectra that we obtain for each peptide will increase in mass, as seen here, where the isotope envelope shifts to higher masses. We can now repeat these experiments a number of times using various incubation times, and what we end up getting is these deuterium uptake plots that you see in the bottom left-hand corner, which is typically a sigmoidal plot. The plot shows deuterium incorporation as function of a log time scale, and the time scales could vary from seconds to a couple of hours or days, as long as days. This is HDX kind of in a nutshell. Next, I will show a general stage of this HDX experiment, how we interpret deuterium uptake plots. This is an example of an epitope mapping experiment that is typically performed quite routinely in a discovery lab. We first look at the feasibility of the digestion of the antigen in water using the same pH, temperature, and conditions that are typically used for also the deuterium exchange experiment. And we perform LC-MS, and we then perform HDX in stage 2 and 3 under the same conditions as the feasibility experiment, but in the absence as well as the presence, now in this case, the binding partner, which is the antibody. We can then determine the regions of the antigen that have stable hydrogen bonding by looking at the deuterium uptake plots. Here are the results where we are performing epitope mapping for 3 ligands. The epitope mapping is displayed as a heat map showing the extent of deuterium uptake as a free energy change. So 1 to 3 are significant changes in deuterium uptake, almost 2 to 3 kcals per mole in free energy is seen for the first and the third ligand of these residues. And we are comfortable that these are epitopes. In region 4, we do not see any differences in deuterium uptake, or it is significantly slow. All the regions in gray color are non-observed regions where the deuterium exchange can also be too slow or too fast. Over here in 5, a significant shift in deuterium uptake of about 2 kcal per mole in free energy is seen for only the second ligand, not the first and third ligand in this patch of residues. And we are comfortable that these are residues that comprise this epitope for ligand 2. Finally, we see deuterium uptake, but there is no significant change in deuterium uptake to call these regions epitopes. And they are all regions in yellow. Now that we have some background on how we interpret these uptake plots, I will show you and walk you through the results we obtained for the bispecific antibody dimer. So as I mentioned, the first step of the experiment is to evaluate the digestion efficiency. In any HT experiment, digestion and LC separation are done at low temperature and low pH to reduce back exchange. And we use pepsin FPX2 mixed bed column and digest the bispecific antibody. And we do that in this core box. And in this particular experiment, we get about 100% sequence coverage. Then we perform the HTX experiment on the monomer. In this case, the purified monomer. And what you're seeing is the heat map coverage of the regions in blue. The exchange is very slow, and we do not have any observations. Conversely, the regions in red show very high exchange, and there are regions that are highly unstable. All other regions that are in between these are regions where deuterium uptake plus shows sigmoidal behavior. Now we perform HDX experiment on this bispecific antibody dimer in a similar manner. And what you see is a heat map of binding free energy change between the bispecific antibody monomer and a dimer. So this is delta delta G, or the free energy change. So what we see are regions colored in gray that have no observations. And then we also see large number of regions in yellow, or in other words, regions that show there is no difference in free energy change of the hydrogen bonding stability between the monomer and the dimer. Then we also observe this region, peptide in 52 to 53 amino acid, that show a very small change. We believe this to be very insignificant. Based on all these results, we can generate a new hypothesis on how the dimerization may be taking place. We can propose 2 types of dimerization. One where dimerization results in dimer formation directly through a direct interaction of the scFv arms of the 2 antibodies. And the second case where the scFv arms have an open form that can domain swap with the second scFv arm on another antibody. If a direct interaction takes place where the 2 purple domains for this contacts are formed, the HDX pattern should have slowing down of the dimer compared to the monomer, or regions of contact are stabilized by hydrogen bonding. On the other hand, if domain swapping took place, the contact points in the 2 monomers are identical to the domain swap dimers, and we should not see a major difference in hydrogen bonding in the monomer and the dimer. Based on our results, where there is no change in the HDX pattern, support a domain swapping mechanism. In conclusion, I hope I was able to demonstrate the importance of structural MS and what structural MS methods brings to the table in early development. I showed how we used multiple technologies to understand how the structure of a bispecific antibody that was prone to aggregation as well as dimerization. We looked at its structure with native SCCMS to obtain stoichiometry. And then we extended these studies combining native SCCMS with ion mobility, which is a gas phase separation. And then we looked at subunits and do subunit analysis to take a step further to look at native conformations in the gas phase and narrow the binding regions to the SAFE arms. Finally, I showed results from two footprinting methods, HRPF and HDX, and how we can use these tools to understand the binding interfaces to narrow regions of the sequence that are involved in their binding. These experiments are still ongoing, but our current understanding is that the SAFE binding happens through a domain swapping mechanism. Finally, as we continue to explore new modalities, I believe that structural MS tools will have its own place and will play a significant role in better understanding our molecules. We hope early investigations will improve overall operational efficiencies and bring time to value as shown here. The future efforts are listed here. I did not have time to present our exciting work on the computational analysis that we feel is going to help our understanding and design molecules with better quality attributes. So these include molecular dynamic simulations and molecular modeling, and combining hydrogen high order structures with high electron microscopy and other high resolution separation methods will also provide additional information, and we are strongly pursuing these other areas as well. With that, I would like to acknowledge all the contributors at Janssen and our research partners and collaborators. Especially, I would like to thank Emily from GenX, who did all the HRPF work, Yoshi, who did the HDX experiments, Michael from Janssen, as well as Alex and Sanjay from Protein Matrix, who contributed to this presentation. Finally, I appreciate your time, your participation, and I would like to open the floor for any questions, comments, or suggestions. Thank you.